please take over from here.
Great, thank you. Hello, everybody. My name is Cleo, and today's talk is titled Convergent Evolution through Recurrent Rearrangements in the Primate Amylase Locus. And as you can imagine, I will be talking about the amylase, So amylase is the enzyme responsible for breaking down starches, and it was the first enzyme to ever be discovered in humans in 1831. And although it's very well studied, very little is known about the structure. The amylase gene that encodes for the amylase enzyme is related to diabetes, metabolic syndrome, starch perception, even Alzheimer's. And we do know that there are all of these implications and associations. And as early as the 1980s, we've known that the gene might be copy number variable, but it wasn't at least 2005 that we understood the extent of the copy number variation, and earlier this year, we managed to reconstruct the structure of the amylase locus in diverse human populations worldwide. But this variation in the amylase locus is not only present in humans. We have copy number variation in dogs, pigs, mice, and a lot of other species, and it still remains uncharacterized in a lot of these organisms, including primates. So today I will be focusing in primates, mostly non-human primates, where here we analyzed 244 non-primate assemblies. And from this, we could recover the continuous amylase locus from 70 of them. That means that we could reconstruct the amylase locus in a single contig or a single scaffold, and reconstruct the evolutionary history of the locus and understand the different rearrangements that have occurred. So we were able in total to determine 15 distinct rearrangements that took place in the primate lineage. Some of these rearrangements have never been identified before, like duplications in lemurs or New World monkeys, and some others they have been previously noted but not characterized or elucidated in depth, like the duplication in the ancestor of Caterini that the single amylase gene duplicated into two amylase genes here. So I will be focusing primarily in the old world monkeys, but in order to do that, I want to give you an overview of the structure of the locus and why we're interested in the amylase locus and the complexities and the copy number variation. So the ancestor of Caterini had, like we just mentioned, two copies, an AMY1-2 and AMY1 prime and AMY2B. The AMY1 prime is duplicated from AMY2B, which is the ancestral gene. Similarly, in rhesus macaque, we identified three genes, AMY2B, AMY-M, and AMY1 prime. AMY1 prime is one-to-one ortholog to the AMY1 prime in the ancestor of Caterini, and AMY2B similarly is a one-to-one ancestor to the ancestral gene, while the AMY-M gene is a novel gene. The same pattern holds for olive baboons, where we have the AMY2B, two novel genes, and the ancestral AMY1 prime. And in humans, we have a slightly different situation. Again, we have a lot of copy number variation across different populations in humans, but at least in the reference genome, we have two AMY2 genes and three AMY1 genes. The AMY2B gene is one-to-one ortholog to the Caterini, rhesus macaque, and olive baboon AMY2B gene, while the AMY1 genes and the AMY2 genes are more similar to the AMY1 prime, but they're not one-to-one orthologs. In fact, the AMY1 prime is a progenitor of these genes. Another thing that is quite interesting is that we know that in humans, the AMY2 genes have pancreatic expression, while the AMY1 genes have salivary expression, and the same is not known about the old world monkeys. So in order to reconstruct the evolutionary history of the locus in the macaca, in rhesus macaques, we looked into the syntonic relationship between the six different macaque species and trying to understand and date the duplication, but also elucidate the mechanisms behind it and how the rearrangements occur. So we were able to determine that the duplication is present in the Seneca and fascicularis groups, but is absent from the Silenus group. So this would place the duplication event sometime 4.5 to 5 million years ago. Also, we were able to reconstruct the mechanism that generate these breaks, and these breaks are and the rearrangements in this extent. And the mechanism behind these duplications is a non-homologous, non-allelic homologous recombination event, where this new gene that forms this whole segment that is being duplicated is a mosaic or a chimerism between the two ancestral copies. So part of the sequence will be AMY1 prime-like, and the other one will be AMY2B-like. And also we were able to determine that the breakpoint is right before the coding sequence, and the coding sequence is inherited intact and is identical, almost identical to the AMY2B gene. The same was done for olive baboons. In this case, we were not able to compare it to sister species because the assembly of Papio is a little bit problematic in the region, but we used outgroups. In this case, we could compare it to geladas and other species from the old world monkeys that they all carry two groups, and we identified these two novel genes. So these two genes, we coined the terms AMYP1 and AMYP2. AMYP1 is the first duplication, and AMYP2 is the second duplication that occurred. So we identified that these duplications happen in a stepwise manner. The first duplication, the AMYP1, is shared between Papio nubius and Papio, which I cannot show here because of the complexities with the assembly. And this would place the first duplication event approximately 2 to 4.5 million years ago, while the second duplication is olive baboon specific, and therefore the dating would be approximately 1.85 million years ago. These are distinct duplications from the duplication in the macaque. They are lineage specific, and the second duplication is space specific. And again, NHR is the predominant mechanism that generates these breaks. So all of this is very interesting because we do have in 10 million years of evolution in primates at least 4 new rearrangements that have occurred, and we are trying to understand better the implication of transposable elements or the complexity of the locus, or whether there is some recombination hotspot that generates these breaks recurrently. So we are, I think, in a good way of better understanding this. But there is another question that remains open, and this is what happens in terms of the functional consequences of all of these duplications. And to do that, we use transcriptomics data from the pancreas and salivary glands for these two species. So we have the pancreas and parotid sublingual submandibular glands for olive baboons and rhesus macaque. And as we can see here, the expression in the pancreas in rhesus macaque is primarily coming from the AMYM gene, the novel gene, while the expression in the parotid and submandibular is primarily contributed by AMY1 prime, while the sublingual is more of a composite of the 3 genes. Similarly, the sublingual for olive baboon seems to be the total expression in the tissue of the amylase in the tissue seems to be a composite of all of the olive baboon genes, while we do observe a different pattern for the pancreas, where we have primarily AMY2B expression, similarly to humans. And again, the parotid here is more AMY1, is the contribution of AMY1 prime seems to be predominant in the parotid. And to quantify this further and try to understand the implications, we performed differential expression analysis within species but between tissues. So here we are comparing liver, parotid, submandibular, and sublingual glands to the pancreatic expression. So any log2 fold change higher than 1 would indicate that the expression is higher than in the tissue we are studying than in the pancreas. The control is the pancreas. So we're comparing everything to the pancreatic expression, while if the log fold change is smaller than 0, that would indicate that the expression is higher in the pancreas. And one of the patterns that emerges here is that the lysine in its cluster in each species seems to have higher salivary expression. This is true also for humans, although in humans we do observe a different pattern. It's exclusively salivary expression. Like I previously mentioned, in humans, we do have all of the AMY1 genes being expressed in saliva, while the AMY2 genes are exclusively expressed in the pancreas. In the olive baboon and rhesus macaque genomes, we do see a trend for higher expression of the AMY1 gene in the parotid tissue, in particular in olive baboons, and parotid submandibular tissues in rhesus macaque. But we don't see this exclusivity tissue-specific expression like we see in humans. So if we assume that the or that the human has undergone a subfunctionalization event, I think that it would be very interesting to better understand this, as we might be capturing the subfunctionalization process. So again, putting everything together, now we know that the duplications have happened, the mechanisms behind these duplications, and also we know the tissue-specific expression for all oligobaboons and rhesus macaques. And putting all of this together, we have a more comprehensive story. But looking into the evolutionary history of the locus requires also selection analysis. And to do that, we took all of the sequences of the primate amylase genes and looked for selections that might be under positive selection and episodic diversifying selection. We identified 6 sites that they're under episodic diversifying selection and 11 sites that they're under positive selection. And also we annotated active sites, catalytic binding sites, calcium binding sites, glycosylation targets. So all of these are sites that they are important for the function of the amylase gene. And we do find nucleotide codon substitution that is present in all bird monkeys and overlaps with an active site, so there could be implications for that. So we've modeled this with AlphaFold to try to understand the changes in the structure, and we're also looking further into understanding the implications of this. Moreover, we are looking into transcription factor binding sites that reside within the promoters of all of these genes and try to do quantification of the different transcription factor binding sites and how they relate with a gain or loss of expression in a particular tissue. And also putting this in the big context of rearrangements and structural variation, I think it's quite interesting to understand how structural variation as a force of genomic innovation can rewire the genome. And now put transcription factor binding sites in close proximity with these novel genes and therefore result in new functions or novel expression. And with this, I would like to thank everybody from my lab. This is us putting our amylase to use. The Omer, Gokchman, and Stefan rule that they participate in this project. The funding bodies is NIH and NSF, and the organizers for giving me the opportunity to talk here. And I'm happy to take any questions. Thank you.
Great, perfect timing. We have a few minutes for questions. Any question from the audience? Any question here? All right, I guess I'll ask a couple of questions to get started. First, in terms of term you use, is this example of sub-functionalization or new functionalization? Sorry about my timer.
Yeah. So I think that this is something that we're always thinking about in terms of gene duplications, right? There are 3 pathways like pseudogenization, subfunctionalization, new functionalization. There is actually a really nice paper in Heredity from 2011 from Matthew Hahn, where he explains the terms quite in depth. I think it's also interesting because amylase is the classical example of dosage effect because the copy number variation in humans has a distinct dosage signal. It doesn't have distinct functions. But I think in this case, with the primate amylase in comparison to the human amylase, these genes still have all of these functions in the pancreas and the parotid, sublingual, and submandibular tissues. And then what happens is that they gain specialized expression in one tissue. So I think that I would make an argument for a subfunctionalization rather than neofunctionalization because the function was already present there, right? But it's interesting to see how they evolve. There are more data that I haven't had the time to show here. So we do see that one of the genes already in Papilla nubis that I had it with dust lines has already gone under pseudogenization. So it's kind of interesting because the expression of the gene is expensive for the organism. So unless there is a particular function or a need for it, it will become pseudogenized or utilized in a different way. So there is definitely more to understand here, both from the functional and evolutionary standpoint.
Right. So have you seen any case of pseudo genes?
Yeah. So
and what happened there is
this is actually a
promoter?
pseudogene. Or it's annotated as a pseudogene. I could recover all of the with Exonerate, I was able to recover all of the exons without any stop codons, but all of the contemporary annotation tools categorize it as a pseudogene. So I'm trying to understand it a little bit better.
Okay. Now Lara has a question. Go ahead, Lara. Let me unmute you.
Hi.
Yes, you can unmute yourself. Yes, your host. Of course. Go ahead.
Yeah, I was wondering, so I can see here in your diagram that sometimes you have genes that are like in opposed directions. Have you observed any changes in expression due to that?
So these are the human genes, and I recently published this manuscript on the human amylase, the evolution of human amylase, and we do have salivary expression data. The problem is that the individuals that we have salivary expression data from, we don't have the genomic sequence, and there is so much variation in humans. Some individuals might have from 2 copies in diploid cell, while others might have 22. We also have a manuscript onto that, like the population that has the highest copy number variation. These are Peruvian, spoiler. But the thing is that unless we have the genotype and the phenotypic data in this case being the expression data, it's very hard to understand it. My hunch is that I think that the inversions, especially in the human amylase locus, might be more interesting than the duplication events, because every time we have a duplication, these 2 genes are duplicated intact. So they're duplicated together and being deleted together as a single Mendelian block. So I think that the copy number, yes, is important, but all of these inversions that happen in between the genes and affect the genes, I think they also affect the regulatory sequences, and therefore it might have implications for the expression. But it's something we're actively pursuing, and I don't have a conclusive answer.
Okay.
All right, great. Thank you very much, Cleo, again for your wonderful presentation. I guess it's time to move on to next.
I can't stop sharing.
Okay, I think once the next speaker starts, so it will automatically replace your sharing. Our next talk is from Maria Tocora. Hope I pronounce your name properly. From University of Toronto. I believe this is a recorded talk, but I think she's here to answer any question at the end of the talk. All right, let's go ahead.
Just let me know if the sound's okay. Everyone, this is Maria Tocora from the University of Toronto, and for today I'm going to share with you all my research on the genomic divergence of obligate bacteria associated with an endemic ant species in the Galapagos Islands. So I hope you enjoyed the presentation, and I'm looking forward to answering your questions. So endosymbionts have explored the diversity of insects by opening otherwise non-accessible ecological opportunities. In nature, we have a wide diversity of these interactions, including ants that interact with multiple bacteria species, or even other host species that do not interact with many bacteria. So in the interactions, we can also find facultative or obligate symbionts in which we have these bacteria residing in specialized organs in something called endosymbiosis. With the endosymbionts, as they reside inside the host, we might expect the bacteria being exposed to what is called the rabbit hole, where we have these different selective pressures within the host, changing or affecting the genome of the bacteria by first affecting the genome size or also leading to the accumulations of different mutations. In an evolutionary perspective, lineages that have acquired symbionts are supposed to be exposed to more shared evolutionary history between partners in comparison to other lineages that don't interact with symbionts. We might also expect having a phylogenetic congruence pattern between these partners, where we have the phylogeny of the bacteria mirroring the phylogeny of the host, suggesting shared evolutionary history between these 2 organisms. However, something that we don't know much about these interactions is how these bacteria evolve in concern with their partners across space. And this is actually one of the main goals in my research, to understand how these 2 partners evolve. So I studied the obligate endosymbiosis between ants from the genus Camponotus that is a really widespread genus with more than 1,000 species distributed around the world. And these ants interact with these Blockmania bacteria, and they have been interacting with for more than 50 million years ago. And this bacteria is involved in different metabolic processes, such as development and nutrition. In the Galapagos, we have 3 species, 2 endemic and 1 introduced species. And one of these endemic species, the target of our research, is Camponotus planus. These species have been described more than 100 years ago. And for these species, we have described multiple subspecies, differentially distributed across the archipelago, and with non-overlapping distributions, except for 2 subspecies distributed in the same island of Santa Cruz. Originally, these subspecies were described based on morphological data. However, my preliminary data suggests high similarity across populations of Camponotus planus differentially distributed in the islands. So here you might guess that getting genomic data is essential to understand any patterns of variation, morphological, genetic variation of these species across this distributional range. Also, genomic data is important to understand the evolution of this interaction between Camponotus and Blockmania across the Galapagos. So we went to the islands and we collected samples of Camponotus planus across 10 islands. And collecting these ants was actually a challenge because these ants really like residing in the roots of endemic plant species in the archipelago, especially cactus species, and really like branching across these different endemic plants or even residing inside the holes of volcanic rocks. So finding the specific place where to find a colony is hard, but we were able to do it and collected a couple of colonies that were used in these different downstream analyses that you are gonna see. So now that we have the samples, our next step was to collect and to have our genomic data. So first, we got the de novo reference genome for a Camponotus neotropical species. So previous to our research, there were a couple of genomes available in databases, but none of them were from neotropical Camponotus species. We got the first one for a neotropical species that is actually endemic to the Galapagos. So this genome is composed by around 26 scaffolds containing approximately 95% of the genome. We also annotated the genome, as you can see, in these different scaffolds in the x-axis, and we assess the proportion of codifying sequences in the y-axis, proportion of repetitive and transposable elements, and also the proportion of GC that varies across these different scaffolds. Really important information to be used for other comparative genomic analyses with other species. We also use the genome to understand how different our genome was from other genomes published. And here you see an example with the genome Camponotus JDM, a species of Camponotus sequence from the Mandy Lab at Texas State University, where you see the presence of highly synthetic regions across our genome in comparison to this other genome from a Nearctic species. We have some regions that are no synthetic regions that might suggest some inversions happening across these genomes that we are comparing. Our next step was to get a genome from the bacteria species. And this is important because our question, as I repeat across this presentation, involved 2 partners. So here we got the reference genome for the Blockmania species interacting with Camponotus planus. This genome is around 700,000 BP, and it's actually quite small in comparison to other Blockmania species, as you can see here in this graph, where we have the genome size in the x-axis and the proportion of GC in the y-axis, and we have multiple Blockmania species interacting with multiple host or ant species. So here our genome from Blockmania planus or associated with Camponotus planus is smaller in comparison to the other ones. So our next step was to understand how our samples are related to each other in order to use this information for downstream analysis such as population structure and phylogenomics. So we assess relatedness across Camponorus planus populations by using allele frequency-free methods, 3 methods that I'm going to explain in these graphs. So in panel A, we see one of them that is kin robust, and these represent the fixed difference and shared heterozygous sites across our samples. Each dot is a pairwise comparison from samples coming from the same colony or intra-colony comparisons, nearby colony comparison here, or inter-colony comparisons highlighted in orange. So here we see that values around 0.2 represents full sisters that is expected for intra-colony comparison, and it is actually what we are getting. Values below 0 represents pairwise comparison coming from highly structured populations. That is expected here with samples coming from locations more than 10 kilometers apart, or even samples located in different islands. So here we have in panel B, 2 statistics that is R0 in y-axis and R1 in x-axis, and R0 represents the ratio of fixed differences between individuals, and R1 represents the ratio with shared heterozygosity between individuals. Values near to 0 for R0 represents the closely related individuals, and higher values of R1 represent the more related the value, the higher the value, the more related the samples are. So here we see that our intra-colony comparisons have these values of R0 and R1. So now that we know that our colony structure is composed by full sisters and also by a monogame colony, that basically means having only one queen based on our ecological observations, we then perform analysis based on the population structure of these 2 partners. So here in panel A, you see the genetic structure of the partners by having these different ancestries represented in the Galapagos map. So we know that ancestries are shared across closely located samples, such as these samples from Fernandina and Isabella, or even Santiago and Marchena, 2 closely located islands. But we also see different ancestries coming within islands, such as this ancestry in the north of Isabella or versus the south of Isabella, or ancestries coming from the north and south of Santa Cruz Island. So we have this variation in ancestry across islands and within islands in even different K values that we analyze, such as K6 or K equal to 10. When we got the principal component analysis, we also got different clusters of islands that are closely located to each other, such as these islands located more to the center of the archipelago, or this island that is apart from other islands, such as Floreana, that have only its samples clustering together. We also have samples from Fernandina and Isabella clustering together, and Marchena and Santiago clustering together, islands that are closely located to each other. This pattern was found for hosts and bacteria as well. That basically says to us that we have genetic structure, and this genetic structure is consistent with the geographic distance across our host, but also bacteria samples. So then we analyze the phylogenomics of this interaction by basically obtaining phylogenies for both partners, for the ant on the left side, and for the bacteria on the right side. And what we see here are different clades that corresponds to actually different islands. So these clades are sister to each other, and when we go to the map, those islands are located close to each other. So here we see that there is, when we compare these different branches, we see that there is a mismatch here within these clades with samples coming from Floreana and from samples coming from San Cristobal. This mismatch might give us some insight on potential phylogenetic congruence coming from these 2 partners, and this phylogenetic congruence that is quite interesting because in previous research that explores the evolution of between host and endosymbionts, generally the pattern is that you find congruence across the phylogenies. So this is interesting that we found this for these specific populations of ants and bacteria. So to conclude, our main question was to understand how microbes evolve in concert with the partners across the space, And we found that the bacteria strains genetic distances correlate with the host genetic and geographic distances. That is quite interesting. And we also found lack of phylogenetic congruence across these two phylogenies that we analyzed. In our next steps, we suggest getting more sequencing, more sampling, and also studying other Camponotus species, such as Camponotus massilentus, that is an endemic species, and Camponotus zonatus, that is an introduced species. Here in the map, these bluish regions, zones, are areas that haven't been sampled, so we need more samples from these specific localities. This work is a joint collaboration across different institutions from Canada and Ecuador, and I really appreciate everyone's help, especially the rangers from the Galapagos National Park, who were a key part from our team fieldwork. And please stay in touch with me with my research. You can contact me with any inquiries to my email, and also to get access to my code and my bioinformatic analysis on my GitHub. Thank you so much for your attention.
All right, we're at a 2, but if Maria is here, maybe we can entertain one question. Any question from the audience? No? All right, so let's move on to the next talk. From Lara from University of Sheffield.
Hello.
Go ahead, Lara.
Hi, my name is Lara, and today I'm going to talk about recent work that we published in bioRxiv. So if you want to check it out, the link is here. And it's titled Gene Turnover and Contingency Facilitated the Repeated Evolution of C4 Photosynthesis in Grasses. So first of all, I want to just introduce what C4 photosynthesis is. It's a carbon concentrating mechanism that makes plants more efficient in hot and dry environments. And it consists mainly just in spatial separation of the metabolic reactions of photosynthesis between two different cell types. For this to happen, we need both biochemical and anatomical changes. So for example, in terms of anatomy, we have here rice, that is a sectioning of a rice leaf and a maize leaf. And we can see huge difference mainly in number of major and minor veins and the size of this specific cell type that is called bundle sheath cell. This is important because in terms of biochemical changes, what happens is that in C3 plants, all the different reactions happen in the mesophyll cell mainly. But in C4 plants, we have a first fixation of the CO2 in a mesophyll cell, but then the corresponding organic acids move to a different cell that is the bundle sheath cell, that are these cells here. And most of the important primary photosynthetic reactions happen in this other cell type. So this C4 cycle evolved convergently more than 60 times in plants, and specifically in grasses, that is the system I work with, we had about 20 origins, and they are all clustered in one clade. So in grasses, we have, this is a grass phylogeny, and we have two main clades, the PACMAT clade that includes all these subgroups, and the BOB clade that includes all these subgroups. Both of them contain important, very well-known crops, and the main difference among them is that the BOB clade contains only C3 species represented in gray, whilst the PACMAT clade has C3 and C4 species in most of their subfamilies. So something that we thought, our hypothesis is that for some reason, the PACMAT clade carries some precursors that act as genetic enablers to facilitate C4 evolution exclusively within this clade. And the aim of this work was to identify gene gains or losses that might represent pre-adaptations to C4 photosynthesis in the last common ancestor of the PACMA clade. And these genes could be either gene losses at the base of the BOP clade or duplications and de novo gene birth at the base of the PACMA clade. So either losses here or gains here. And most of these groups have some representative species sequence already. So we can see here, for example, in the Panicoideae, we have 41 species that are sequenced out of 3,500 more or less. Similarly, in Poideae, for example, we have 47 out of 4,000. However, in the Aristidoideae group, here at the top, we have no representative sequence. And this is a very important, very relevant group because it's the most divergent group within PACMA. So it's this clade, this subfamily, is sister to everything else. So to really account for variation that happened in this branch, we need to have some representation from this group. So the first thing we did was to sequence two C4 lineages, well, two species from the main C4 lineages within Aristidoideae. And these are Aristida abstentionis and Stypograstis certigluma. Aristida is diploid, whilst Stypograstis is tetraploid, and that probably explains why the Stypograstis genome is a bit more fragmented, but they are both quite complete. So we see the BUSCO scores are pretty good. So for our purposes, even though it's a bit fragmented, they are good enough. So what we did with these genomes, along with another 32 RAS genomes, was to use AutoFinder to classify all the coding sequence genes in orthogroups. And then AutoFinder reconciled the gene trees with the species tree and identifies duplications in the different nodes. So we were able to place almost 91% of the genes in orthogroups. This is the species tree for the species we used. And these numbers here are the numbers of duplications identified in each node for using AutoFinder. And the most important for us is this one here. So this is the base of the PACMA clade. And we can see here 482 duplications. Important to note that this is not a burst of duplication. So the rate is similar in all the branches when we account for the divergence. So using also those results, we identified not only the duplicated, but also the de novo genes in PACMA and the gene losses in BOP. To do that, we just looked for genes that are, for example, present in PACMA, but absent in both BOP and outgroups. So we would consider that the de novo genes, and also genes that are absent, totally absent in BOP, but present in the outgroups and in PACMA. And those would be gene losses in BOP. And in total, we got about 1,000 candidates that could be precursors. We also define a core set of candidates that are the dash fractions, and those are the ones that are retained in all the C4 species from our analysis. For example, in the duplicated, these are the orthogroups that contain genes that were duplicated and retaining all C4 species. And here, we have just the copy number variation for those orthogroups. So we have a few transporters, a few enzymes, and a few other genes with diverse functions. And we can see, we could see that the number of genes in the PACMA clade is usually higher. So I checked while accounting for ploidy level, whether the gene copy number differences are significant. And we saw that in most cases they are. So we have a significantly higher number of genes in these orthogroups in PACMA when comparing to BOP. So we assume that this means probably multiple duplications and reduced gene loss in these orthogroups. for this core set, we also verified the duplications using independent phylogeny. So we constructed our own trees, and we found that, yeah, we have in all of them, well, in 15 out of the initial 18, we have 2 genes in 2 copies in PubMed, and only 1 copy in Bob and 1 copy in the author group. These are 2 of our preferred candidates, but yeah, they are just an example of the work we did. So of course we looked at the genes that are well known for C4, so the core C4 genes, and we only found 1 gene that was duplicated, that is this carbonic anhydrase, and this is an enzyme that is the entry step of the C4 cycle. So it performs the initial fixation of CO2 into bicarbonate, and this happens in different cell types in C3 and C4. It happens in the same cell type, but this carbonic anhydrase is a family with a multi-gene family, and only one of the lineages has been co-opted for C4 cycle, and this is the one, so this lineage is the one that has been duplicated. This has been reported before, but we confirmed that the duplication was at the base of PubMed, because we now have these aristidoid genomes that we didn't have before. So the other thing we did is for the whole set of genes, we looked for significant Go terms, and we found only in the duplicated genes, we found a few overrepresented Go terms, and they were mainly related with transporter activity and catalytic activity, mainly oxidative stress. So just to have an idea of what we found, the most important terms, or the ones that we could clearly or more clearly relate to C4 cycle, were the organic acid transport, amino acid transport, and the glutathione metabolic process, and this kind of oxidoreductase activity. So these would be involved either in transport of molecules that we know that is important for C4 evolution, because all these acids has to be transported again between different cells, and in redox, that is also important because photosynthesis generates oxidative stress, and if you're working a different cell, you have to manage that oxidative stress in different cell types. We also looked at differential expression between mesophyll and bundle seed cells in using published data in maize and sorghum, that are C4 crops, and in rice, that is a C3. And we found that sometimes for a few of our candidates, we find a shift in gene expression. So for example, this gene is preferentially expressed in mesophyll in rice, but it shifts to bundle seed cells in the C4 crops. And we found that for these 2 genes and a few others. We also found some genes that are not significantly, not differentially expressed in rice, but are differentially expressed in the C4 crops. So we think that these are candidates that could help, could add variation that can be later co-opted for the C4 evolution. So just to finish up, take-home messages. We generated 2 new aristidoid genomes together with other publicly available rat genomes. We used them to identify genetic precursors that are present in the PubMed clade and absent in the Bob clade that could enable the evolution of C4 photosynthesis. These candidates are mainly involved in carbon metabolism, amino acid and nitrate transport, oxidative stress management, and transcription regulation. And we think that the repeated evolution of C4 photosynthesis in PubMed grasses was not driven by a single genetic event, but rather by the opportunistic utilization of pre-existing genetic diversity. Thank you for your attention, and I would like to acknowledge my supervisors and colleagues, especially the PhD student in the lab, Hamed, that is a co-author, a co-first author in this paper, and of course the rest of the Dunning lab. Thank you very much, and thank you for your attention, and I'm happy to answer any question you might have.
All right, great. Thank you for the presentation. We may have time for a couple of questions. Any question from the audience? I'll get started by asking one. Have you looked at the mechanism by which the novel genes were created? Was there any mechanism that's more important than the other one?
Sorry, can you repeat, please?
So have you looked at the mechanism by which the novel genes were created? Was there
Not,
any mechanism that's more important than the other one?
yeah, not yet. That's interesting. We should do that at some point. For now, we just kind of went through the main or to find the results to define the genes, but we haven't looked at how the genes were formed, not yet.
Okay. Have you also looked at the content of transposable elements and the other repetitive sequence, the other tandem repeats in your genome?
So,
Okay.
no, that is kind of a bit out of the scope because we were focusing mainly on the coding sequences. You know that in grasses there's bursts of transposable elements, and I'm sure that to some extent that's also part of the evolution of C4 photosynthesis, but I think it's much more difficult to account for it because we don't know. Yeah, we cannot define what the transposable element is doing unless it changes the expression in some of the important genes. So that's something we could look at. We haven't yet.
All right, great. Thank you, Lara, again for the presentation and I think the questions. And for the time concern, let's move on to the next.